we were talking about is this part, which is directly from that Nature Genetics paper about measurement model and expression model. If you recall, we covered that one. The Poisson is used as the measurement model, which means that we consider lambda GC to be the true expression value of gene G in cell C. But we don't get to measure this true expression value because we're doing sequencing. Sequencing as itself is a sampling process, right? So we said that sampling process means in this case, we can simplify to be, okay, you have many, many genes with many, many cells. Their true expression are in RNA molecules, and they are competing for sequencing reads. So therefore, we consider the sampling process to be a multinomial distribution because the total number of sequencing reads will be allocated to genes in cells based on the true expression value. And we also said that Poisson is an approximation for multinomial distribution. And yeah, so I won't go into detail here, but basically because of that property, it's reasonable to assume the count you measure, the UMI count you measure for gene cell C follows a Poisson distribution. And that's the measurement model. So lambda GC is the true expression level. From that, you do Poisson sampling, you get a count. And then the expression model is about how lambda GC is distributed. And in problem 1 over here, we assume lambda GC to be a constant alpha G for all cells, but it's only specific to gene G, which means that we assume for the same gene, its true expression level in all cells are equal to alpha G. That's what we mean. So I think in terms of data generation, this actually specifies a data generation process, which is what we imagine to happen in a real biological experiment. That is, you first have every gene G expressed at different levels, lambda GC in different cell C. That is the biology. And from that, you sample counts, and that's the sequencing. So measurement model is for the sequencing step. Expression model is for the underlying biology, is for the variation of one gene G's expression level, true expression levels in different cell C. Does that answer your question? Yeah, but I think what you said, something like thresholding indicator, I think that was a misconception of the point mass. Yeah. Yeah. So remember in our lecture notes, I think in one of the previous slides, when I talk about negative binomial, I talked about 2 ways of deriving the negative binomial. The standard or the simpler way is to think about coin tossing, right? It's the number of successes before we say R failures. That's one way to derive negative binomial. And then the number of successes before R failure follows a Poisson distribution. And then we use the gamma Gaussian gamma Gaussian hierarchical model. Then we can also derive the distribution for the count following Poisson. So basically you have you have the gamma distribution for the lambda, then you have the count from Poisson lambda. Then when you can write down the joint distribution of the Poisson count and lambda GC. And to just get a marginal distribution for the count only, you integrate out the lambda parameter. And we can show that after you integrate out lambda parameter, the probability mass function, if you recall, that's how we specify a discrete random variable, right? Count is a discrete random variable. That probability mass function happens to be the same function as the one you get from coin tossing. So that's what we said is negative binomial. Yeah, so when we say some random variable follows some distribution, one way to say that, or one way to prove it, is to check whether the probability mass function matches the distribution you have in mind. Yeah, actually for this question, Besides what I have in the lecture notes, there should be plenty of online material if you Google negative binomial, and 2 ways to derive it, one for coin tossing, one for gamma Poisson hierarchical model. Okay, and also just as a review, since we're here, basically you see compared to this one, the reason we designed the problem in this way is because when it's point mass, lambda GC has no variation itself given G. But when lambda GC is from gamma, alpha G, beta G, pay attention that these 2 parameters are only specific to gene G. Then you have the lambda GC with variation. So it's no longer a constant. Because of this variation, will go down to your Poisson for generating count. Because now for each, let's call X GC following Poisson lambda GC, their parameters lambda GCs are different. So therefore, ultimately, your X GC obtained in the second way, in this second problem, would have bigger variance than the X GC you obtained in the first problem. Because in the first problem, the variation of XGC only comes from Poisson, but now it comes from both Poisson and the parameters gamma. So therefore, you have bigger variance. And that's why intuitively, you can see why negative binomial has bigger variance than Poisson, because the parameters upper level distribution gamma also contributes to the variance in your counts. Okay. All right, any other questions? Yeah, so I think for questions like this, maybe I would suggest that in the class, whenever you find something you're not clear about, it's much better to ask immediately instead of waiting for the homework. I mean, for the negative binomial derivation as one example. Okay, or you can come back with a question next time. That's why I want to leave some time for Q&A at the beginning of each lecture. Okay, so now let's continue with the lecture on PCA. So last time we spent quite some time in the past 2 lectures on PCA. Hopefully you understand more than before. So how many of you have this feeling? Like after last 2 lectures, your understanding of PCA is more than your previous understanding? Okay, only 1 hand I saw. 2. All right. Yeah, thank you. So maybe we'll talk a little more today. So hopefully we will give you another way to interpret PCA. So just so recall, the 2 ways we give you, one is to maximize the variance in your PC. Second, from PC, like K PCs with K less than P, P is the original feature number, you can best restore the P dimensions from the K PCs. Okay, so yeah, this is where we stop here about how do we choose the number of PCs, right? Because each PC is a meta feature that combines information across of the original correlated features. So how many PCs should we keep? And that's the choice of the K. What's the K PC? So we said that the most intuitive and widely used way is to do the elbow plot. So here the standard deviation is the standard deviation in each PC. So the PCs are ordered from the first one, second one, third one, along the way up to the 20th. So this is the number of the kth PC. And we see that as expected, the standard deviation decreases as the PC index goes. Maybe let me just ask you this question, which is kind of a test on your understanding. How do we calculate the standard deviation? How do we get a y-axis? Based on what we said about PCA calculation. What is the value of the variance of each PC? Yeah. You're close, but it's not correct. It's not 100% correct. So the eigenvalue of the X transpose X, the sample, the covariance matrix, P by P covariance matrix is the variance. So lambda 1 is the variance of PC1. one. Lambda 2 is the variance of PC2. So lambda 1 is the largest eigenvalue of X transpose X. And here the standard deviation is the square root of the variance. So basically we're plotting the square root of lambda I for the ith PC. Yeah, so we can write here. So this is what we are plotting. So lambda I for each the ith PC. And as expected, because our PCs are defined by ordering the lambdas, so that's why it's not surprising at all. There will be a decrease. But what elbow plot means is that you look for an elbow. Okay, where would you say is the reasonable elbow? Any ideas? 7. So someone said 7. 7 is right here. Any other opinions? 9. Okay. Yeah, now we see how people's definition of alpha is different. Any other thoughts? Okay, so some people may just choose 10 because 10 is like the rounded number. But anyway, so I think it's actually either 7 or 10, but they are actually close. And in fact, the Surratt authors chose 10 here. All right, so identifying the true dimensionality of a data set can be challenging, uncertain for the user. We therefore suggest these multiple approaches for user. The first is more supervised. Okay, let me be slow here. Exploring PCs to determine relevant structures of heterogeneity and could be used in conjunction with GSEA, for example. Okay, how many of you have heard of GSEA? 4. Okay, what does it stand for? Yeah, gene set enrichment analysis, short for GSEA. And what's the purpose of it?
Defining like sets of genes that are enriched and some like population of your
Yeah, so then what is the input when you want to do the gene set enrichment GSEA analysis? You remember? The genes of you are interested in. So basically, you input a list of genes with p-values. That's what you mean, right? And usually these p-values are for what?
Like differential expression.
Yeah. So then you input these genes with p-value, like differential expressions between two groups. So potentially the p-values, the smaller, the more interesting the genes are, right? That's what you have. Then when you input this into a GSE algorithm, what else do you need to specify? Yeah, I know we didn't cover this, so that's why I'm just giving a, oh, sorry. So you're saying for each input gene, you need to give the effect size. Or correctional? Directionality. Yeah, but when you have the effect size, do you still need the p-values? No, you only need one of those, right? Because it really depends on what you provide to the algorithm. But ultimately, no matter what you give, effect size with signs or p-values without signs, because p-values are between 0 and 1, you need to give away so your input genes are ranked, right? They're sorted from the top most interesting one to the bottom least interesting one. And what else do you need to specify to run GSEA? For sure, species, right? You need to let the algorithm know what species these genes are, because you're basically inputting gene names. And then what do you expect to get out of it? The output of GSEA. As the name stands, gene set enrichment analysis. No response?
Gene set.
Gene set? Just the gene set.
I mean, gene set.
You need to input gene sets or you will get gene sets out? Okay, but the gene sets are your specification or it's stored in the database or what?
I think it's like definitely. It's possible. For example, if you ask on certain C5, C7. So that's up to you. But in theory, somebody can go into people.
Gene sets. And then, did you raise your hand? Species? Sorry, I think for people who are behind, you cannot hear anything, right? I encourage everyone to speak up. At least you should speak as loudly as I am. Yeah, try your best. Yeah.
I think this
But the differential expressed genes were from previous analysis. It's not from GSEA. GSEA is downstream of differential expression analysis. And here GSEA is downstream of PCA. So I'm trying to help you understand what this sentence means. So I'm actually trying to show you one example, more than one example, but this is one example about how do we do research, honestly. When you read something you don't understand, how would you approach the problem? How would you try to use this, confusion as a way to help you understand? Yeah, because we accumulate knowledge in this way. When we don't understand something, we look it up, then we know more. Yeah, so that's why I think this is a good example, because I'm not sure whether we'll talk about GSEA in this class, but I will just use this chance to introduce it at a high level. At least you know what it's supposed to do. Then maybe, yeah, so the input will be a gene list with some statistic for you to rank the genes. The statistic could be some effect size with signs, right? Means that, okay, the gene is upregulated, downregulated. If it's upregulated, the sign is positive. Downregulated, the sign is negative. And you have the absolute value indicate the magnitude of the effect. That's the effect size. Or we could have just a p-value, because if you don't care the sign, then the p-value would be, I would say, monotone with the absolute value of the effect size. So typically, if you have a larger absolute value, say +5, then the p-value should be smaller than if the effect size is +2. The p-value just indicates the degree or how much confidence you have for rejecting the null hypothesis that the gene is not a D gene. Because under the D gene, under the null hypothesis, you will be able to compute the probability that you get such a effect size or even larger effect size. That's how we define p-value. Okay, so coming back to the GSEA, you input a list of genes with a way to rank them, some statistics. Then what you get out of it is for each pre-specified gene set, you get a p-value. But this p-value is called enrichment p-value. What it means is that whether you have a significant overlap, I shouldn't say overlap, whether the gene set, so we have, say, if the database has 5 gene sets, each gene set has a certain biological meaning about, say, one pathway. So we can interpret this gene set as a pathway. And then we can say, oh, whether the genes in this set are more towards the top ranked genes in my input list. If that's the case, then GSEA will give me a higher score and a smaller p-value for the gene set. That means, oh, I could use this gene set to interpret my list of genes. That's how we use it. So ultimately, the reason why we use GSEA is we want to interpret our genes. We want to know the functions of our top genes, but we don't want to say, okay, we need to truncate our gene list. So we only input the top 10, top 200. We don't need to make that choice. We input a whole list with statistics to rank the genes. And then GSEA will give us the overall enrichment about how this particular gene set is enriched in the top, enriched in the bottom, enriched in the middle. So right where the gene set is enriched will be revealed in the GSEA score. Okay, do we understand this part? What does GSEA do? How many of you have not heard about the word pathway? What does pathway mean? Yes, so we do have people there. So yeah, so I know this is something I actually understand more as time goes over the years of teaching. As an interdisciplinary field, bioinformatics, you cannot assume everyone knows everything because people come from different backgrounds. So what is a pathway? Can someone who understands help explain? Yeah.
So we should call it do some biological function that's related.
Yeah, maybe one famous pathway. Can you? Okay, what does it do? mTOR.
So the growth regulation pathway.
So it's about cell growth. Yeah. So I would say the pathway, you will encounter this term, I think mostly in cell biology, right? Cell biology, biochemistry. You'll see this. Basically, is how one gene regulates the other gene, and this other gene regulates another gene. So it's how does genes regulate each other to achieve a certain function? It's not so basically when she said cell growth, it's not controlled just by one gene. Our biological system is more complex than that. So there are some key genes to play the role. And understanding the pathway is the ultimate molecular mechanism for treating disease. You want to know what genes trigger, say, cancer, melanoma cancer, then probably we can look for some steps in the pathway as drug treatment. So that's how things typically work. I think for students who are interested in biology or bioinformatics, I think this level of understanding is essential, right? You need to know why we care about this. It's not like we're doing this just for the fun of this. We want to make them useful. So therefore, for bioinformatics, I would say any computational methods are tools. Just like experimental methods are tools, right? Single cell RNA-seq technology is an experimental tool. We want to use it for measurement. And then computational statistical methods are tools for analyzing our data, but we need to know the purpose. Why do we want to do this? Okay, then coming back to this, the first is more supervised exploring PCs to determine relevant sources of heterogeneity and could be used in conjunction with GSEA, for example. How do we interpret this sentence? Because here we are trying to determine the number of PCs. Don't forget where we are. So then how do we use GSEA? Can you imagine? I know the authors didn't give details, but now is the time for guessing. What information? Yeah.
So genes that represent how the variances that the most.
Perfect, right? Loadings indicate genes' contribution to this PC. They could be positive, they could be negative, they have signs. So then what you could do with GSEA is that you input all genes with their loadings, say in PC1, as the statistics for ranking the genes. And then once you input into GSEA, you will see what are the gene sets, predefined gene sets, enriched in the positive loading genes, right? Top positive loading genes. What are the gene sets enriched in the most negative loading genes? Then you can interpret those genes. So maybe you can say, oh, PC1 is about this cell growth pathway, then I can interpret it. I do the same thing for PC2 using the loadings, see whether we can interpret. And we stop until the PC's loadings do not represent any meaningful pathways, then we stop. So this is like a biological way of interpreting your results. And I would say GSEA analysis is very common if you are doing anything that involves ranking genes. For example, if you develop a method to rank genes in a way you think is more reasonable than previous methods, like for example, you develop new DE method, then how do you justify the methods doing well beyond your simulation? Because on the real data, there's no ground truth. You don't know what's right, what's wrong. then using GSEA is a way to say, oh, my results are biologically more interpretable, then it's useful. So this is very typical in bioinformatics method papers. Okay, that's the first way. Thank you for the nice discussion. The second, elbow plot. I think there's a typo in the website. The second and third, I think they both refer to elbow plot because the second one just stop here. Second elbow plot, no more. The third is a heuristic that's commonly used and can be calculated instantly. In this example, we might have been justified in choosing anything between PC 7 and 12 as a cutoff. So I think this heuristic is for elbow plot. So that's why I think it's for the same point. So we heard 7. So the website says from 7 to 12 right here can be justified because this is where the elbow seems to be at. Okay, they choose 10 here, but encourage users to consider the following. Dendritic cell and NK something may recognize that genes strongly associated with pieces 12 and 3 define rare immune subset. So the authors is arguing that for this data set, the one on the Sura website, PBMC3K, actually pieces 12 and 3, even though you may decide to throw them away based on the elbow plot, because if you stop at 10, you wouldn't go far to 12 and 3. But the authors are saying that they actually have some genes that are interesting and can help you explain some immune cell types. So, however, these gene groups are so rare, they are difficult to distinguish from background noise for a data set of this size without prior knowledge. While this is the ultimate challenge we face when we do those data analysis, because if you remember, I mentioned in the very early class, I said that the reason we need statistics is because we know our data is noisy and we need to distinguish signal from noise. And we do PCA because we don't believe our data needs such a high dimension, 2,000 highly variable genes to explain. And we think because the genes are some of the genes are correlated, then probably we just need a much smaller number of PCs to explain the variance in our data. So you see here, we stop at 10 because we see that by including more and more PCs, their contributions in explaining the variance is minimum, so we don't go there. However, even those small variants they explain could be biological. That's what this point 1 is saying. But we cannot tell it from data. We have to use knowledge because from data, the variance explained by PC12 is so small, then it's not bigger than the variance we expect from just the measurement model, the Poisson model. The Poisson model itself will give you some degree of variation, right? So that's why I hope you realize that data analysis can only go this far if you just use data, because for using data, no prior knowledge, what you can do is to use something like this to help you decide where to stop. But with biology, with your knowledge, then you have more, prior information, prior knowledge to help you know, oh, there is biology. So that makes the bioinformatics analysis tricky, honestly. And I hope some of you who are really interested in this field understand that knowledge is very important. That's probably your niche. And maybe I can just go beyond this course a little bit to say more about this field. In the bioinformatics field, if you look at researchers, you will see people can belong to maybe roughly 3 circles or even more. But I would just say 3 major circles I know. People who focus on biology. These people are mostly professors, faculty, PIs in the medical school, in the life science department. So to them, like I said, the computational methods are tools to use. They don't really care about whether the method is novel or not because they want to discover new signals, right? Find new cell types, for example, find new marker genes. And the second group of people are method developers. And we have several faculty like this at UCLA. And so for method developers, we look for the limitations of existing methods, and we hope to develop a new method to solve this limitation. So for this purpose, you need to understand what's going on in the existing methods. So you can find their limitation. So like if you have a biology collaborator who told you, oh, GSEA doesn't work well on my data, then you need to find out the reason why it doesn't work well and how can we improve it. So this is the perspective of method developers. But among these people, there are people who are computer science training versus statistics training. I'm just listing two groups. So for computer science training, maybe people focuses more on the algorithm, on the computational efficiency. For example, sequence alignment was a perfect example where computer scientists are very good at, right? How do you fast align your sequencing reads to the genome, to the reference genome? And what we focus on are the statistical aspects. That is, given the data, can I use my statistical model to help me distinguish signal from noise? And that's exactly what that Nature Genetics paper is about. So that's why I like that paper a lot, because I think it's an exemplar. It's the example of how statistics can help with data analysis. So that's why if you want to read one paper among the things I sent, I strongly recommend that paper. Yeah, and it also involves the PCA idea we talked here. So anyway, so I'm saying that you cannot become a method developer just by taking this class, because this class we aim to be broad. We aim to give you some high level ideas. So we hope that with this class you can be a better user, at least, so you can better understand what's going on in the method, decide whether the method manuscript publication was written clearly enough. Because if you don't think so, you're probably right. You're probably that the method wasn't written clearly enough. So you can have more confidence in your judgment. And also maybe it's better, easier for you to communicate with method developers. But if you are interested in becoming a method developer yourself, I think you need more classes beyond this one. So that's what I think. All right, and also I think for people who focus on biological data analysis and the method developers, I think there's great room for collaboration, because even though we are both in bioinformatics, but we focus on different aspects. So in particular, in my opinion, I think if you are interested in becoming a method developer, then the limitations you see in existing methods can be from the analysts, people who use the tools because they understand better where the shortages are. Okay, I hope this can be useful, especially for junior graduate students when you choose your research direction in bioinformatics, whether you want to become a method developer or you want to become a user who can use the tools in a smart way to discover new biology. But I think either way, you need to have your niche, you need to have your expertise, right? I think if you are not understanding the methods nor the biology underlying, then you need to choose one to improve, whether you want to learn more biology or more methods. Yeah. Okay, so we encourage users to repeat downstream analysis with a different number of PCs, 10, 15, or even 50. As you will observe, the results often do not differ dramatically. This is something data set dependent, but it's worth checking. We advise users to err on the higher side when choosing this parameter, number of PCs. For example, performing downstream analysis with only 5 PCs does significantly and adversely affect results. Okay, so this is from the SIRAT. Now I want to introduce this algorithm, which I found useful, called needle algorithm. It's very interesting that this paper was published in 2011 in a computer science workshop. So it's not a journal, but this paper has received a lot of citations, I think because the algorithm is useful. And I was a little surprised myself that this wasn't developed until 2011, because to me, this idea is very straightforward, but I'm not sure whether it was in some old literature, but people just didn't know. Yeah. Anyway, so this algorithm, what it aims to do is to find the knee or the elbow. So basically, if the curve goes upwards, it's called a knee. And if the curve goes, if the curve goes downward like this, it's called an elbow. But you can see the equivalence between the two, because you could flip your y-axis, then this elbow becomes a knee, right? So it doesn't matter. So that's why this idea can apply to both ways. But in a mathematical sense, we call this a what? we call this a concave function, and we call this a convex function, right? So if it's a convex function, we look for an elbow. If it's a concave function, goes upwards, we look for the knee. And this needle algorithm also has this interesting name in the title, finding a needle in the haystack, detecting knee points in system behavior. So what it does is that suppose you have these dots measure, you want to find a knee. And the idea is that you first normalize your y-axis and your x-axis so they both become 0 to 1. You get a square in your coordinate system, 0 to 1 in y, 0 to 1 in x. Then you draw this diagonal line, y equals x, so the straight diagonal line. And then what you did is that for each point, you measure the perpendicular distance from the point to the line. And that's what this dashed segment represents. And because, let me ask you one question, it's a simple math question. What degree is this line? Is for each short segment, what is the degree of it? Relative to the y-axis. Let me just ask this question. Maybe I can make it more explicit. Let's say the degree in the range of 0 degree to 90 degree. What's it? 45 degree. That's right. Yeah. Then, because it's 45 degree intersecting the y-axis, if you extend it, then you see the smaller angle is 45 degree. And then what you did is that you rotate each segment clockwise, so they become vertical. They point down. And then with this, you do what? With this, then you basically do the, we use these measure, we use this, okay, let me just try to say this. A depicts the smooth normalized data with dash bars indicating the perpendicular distance from y equals x with the maximum distance indicated. So it's this point. B shows the same data, but this time the dash bars are rotated 45 degrees. The magnitude of these bars correspond to the difference values used in needle. So basically, once you rotate them here, you will record this bar. And then actually what I'm saying is that you will use this to make this final curve. But I think something confusing here, I want to clarify, is that the height of the bar is the same as this bar. So it's not like once you rotate it, you make it longer, because we know that if you record the distance from here to here, it's longer than the distance from here to here, right? But we didn't change the distance. That's, I think, the confusing part here. But we keep the same distance, we just rotate it. The reason we rotate is to draw this curve. That's it. Basically, you're shifting everything down to the x-axis, so you get this curve. And the reason is that, okay, this is still the maximum as we know here, but the authors has imposed some way to set a threshold, which is this horizontal line here. So the idea is that if this point is the local maximum above the threshold, so basically you see from here to here above the threshold, this is the only maximum, then you record it as the knee. So that's just the last step for thresholding. But I think the most important idea is that you're looking for the perpendicular distance. Yeah. This is the needle algorithm. So we actually used it in one of our recent papers, and we found it to be working very well. So I think I would suggest you to try this on PC as well. And it has Python and R implementation.
Don't use the vertical distance on the
Right.
But if you didn't do that and you used the distance to the 45-degree line, wouldn't that be the same as like rotating the whole curve? Like down?
So I'm saying that you're still using this perpendicular distance. That's what I mean. So this is the distance you are plotting here.
Then wouldn't that just be the same curve, just rotated 45 degrees? It's not the same curve.
That's a very good point. For that, I have to say, I'm not sure. Yeah, I have to, but I think even if it's the same curve rotating down, yeah, but because it doesn't look like so, right?
I think it is the
Mm.
vertical distance, the 45-degree line of the X by the X.
But the thing is that if you are getting the vertical distance, you don't have to calculate the perpendicular distance. That's why I don't think it is, because you could directly get the vertical distance, but you don't need the first step. Yeah, so that's why my understanding of the paper is that they're plotting the perpendicular distance, and this step is just to illustrate how this curve is obtained. Yeah. Because I think the perpendicular distance makes more sense to me rather than the vertical distance.
it has to be created. Otherwise,
Exactly, yeah, yeah,
that's how this.
yeah. That's a good point. Yeah, because here, if you go from here to here, the length is square root of 2 instead of 1, right? So there's like a compression here. Yeah, 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 yeah. Yeah, that's a very good question. So basically, this threshold value is something about the tolerance level. So basically, they want to use the threshold value to avoid some, fluctuation in the curve. If you have multiple local maximum, then which one would you choose? So the criteria here is that the threshold value S can be converted in the way to this horizontal line. So by changing this value to be lower or small, or larger or smaller, the threshold will go up and down. So it's kind of about the local neighborhood size related to that concept. So if you shift it very high, then you will most likely choose this one. But if you make it down and you have some fluctuation here, then you will end up choosing the last local maximum. Yeah, so that's in the, and I think in the paper though, that's the major point of this paper. They have some empirical study trying different values of S. And they found that for different values, it depends on whether they call the data as offline or online. I don't know how many of you have heard that. For offline, it means that you have collected all your data and you are finding the elbow, the knee. For online data, it means that you're continuing your collection of data. So you may have more and more data points along the x-axis. Then it makes the decision of where to stop more challenging. So I think the choice of S is more for the online data because you want to know whether I should stop here or I should collect more data because I'm predicting the future. And for the offline data, I think the author said S equals 1 is the recommended choice. Yeah. Yeah, but I will say this is just something very typical as a heuristic algorithm. There is a parameter you can tune. And how to tune this parameter is another question itself. So that's why if there's no statistical model behind it, or there's no way to justify the theory, then the value choice becomes ad hoc. Yeah, but I encourage you to read more detail into this paper regarding the thresholding value, but it's basically a parameter used in the formula to find the threshold. Yeah, I found it's probably coming up from the author's intuition and also empirical evidence. Okay. All right, so that's about choosing the number of parameters. Well, we mentioned this elbow plot. Then if you want an automatic way to choose it, you can use the needle algorithm. And some of you mentioned at the end of last lecture, there's a Find PC paper in bioinformatics, the journal bioinformatics, the package, our package called Find PC. And in that package, there are 6 methods implemented, but it doesn't include a needle algorithm. And this again says that there are many ways to choose the elbow. Even among yourselves, like your decision might be different, like from person to person. Okay, so we talked about this. Now, for the remaining time, I want to talk more about other dimension reduction methods beyond PCA, even though PCA is the only one included in SURAT. But, when you use PCA in SURAT, you are using center and scaled normalized data. The default is the log1 PCP10K. And the goal is to make the data after the normalization, make the data Gaussian-like for each gene, right? Then PCA is designed for Gaussian-like data. I don't know if I said this enough in the last lecture. The reason PCA is Gaussian is for Gaussian-like data is because Gaussian distribution, this is something good to know, is a very important distribution because in Gaussian distribution, you separate the mean and the variance. The mean has nothing to do with the variance. Mean is just where you shift it, right? You can shift your distribution to anywhere the mean changes, the variance doesn't change. And you can keep the mean, but enlarge the variance. So the two parameters are not interfering with each other. But for Poisson distribution, for negative binomial distribution, you don't have this property. The mean is involved in the variance. So that's why in the normalization step, we hope to remove the effect of mean on variance. That is a property of Gaussian, and then you run PCA because PCA is only dealing with variance, if you recall. We can remove the mean, and after the reconstruction, we add a mean back. So the mean can be removed and add back without interfering with the variance. That's something important. But these authors in this paper, and this is the abstract of this paper. It's called Feature Selection Dimension Reduction for Single-Cell RNA-seq based on multinomial model. They don't want to go through the transformation, like normalization. They want to work with count data directly. And this is how the method is called GLM PCA. So let's read this part in the abstract. Current normalization procedures, such as log of counts per million, this is log 1p CPM. But we said that, okay, or in the paper we give to you, the preprint, it says log 1p CPM is not good, right? It's worse than log 1p CPU 10K. And this is something we hope you can check yourselves in your homework. And just as a hint, it is possible that none of the methods stabilize variance. It's possible. So therefore, if that's something you found, don't doubt. Yeah, it's possible that they're not just perfect. But that's also another your self-confirmation of the normalization may not stabilize the variance. Okay, log 1p CPM and feature selection by highly variable gene procedure. This is exactly what we have in SURAT. Produce false variability in dimension reduction. So then the authors propose generalized, they call generalized PCA. But in fact, the GLM stands for generalized linear model GCA. That's where GLM comes in for non-normal distributions and feature selection using deviance. So deviance is the concept in generalized linear model that's analogous to the variance in linear model because linear model assumes normal distribution while generalized linear model assumes non-normal distribution, which I will briefly cover in this class because I think it's very useful to know. So this is a very important figure from this paper, which is very shocking at the time when it was published. That is, look at the left panel. It's the raw data. It's the UMI count data for one single gene. So we plot the gene's UMI counts in cells in this histogram as a distribution. What does it look like to you? It's unimodal, right? One mode distribution. Looks like negative binomial. Actually, it can be fitted well by negative binomial with a mean somewhere here. But once you do count per million, that is, you scale the genes count into count per million. Remember what we did is that for every cell, we take all the counts from all the genes and scale them together by one scale factor. The scale factor is cell specific. Then we make the genes new total count to be 1 million. And so every gene's counts are changed. And what happens is that 0 stays at 0, but non-zeros are changed into this distribution. So you see the gap between the two. Then you do log 1p, CPM plus 1 times and log 2. You see you have a further separation of the two distributions. And the author said that when people talk about when they talk about zero inflation, when they talk about dropouts, which are dropouts was a term people use for single cell RNA data a lot before. But this paper changed people's view because they're saying that even the original distribution looks unimodal, you could make it bimodal. you will think, okay, this is like a zero mode, this is non-zero mode. Basically, the difference between zero and non-zero is exaggerated by CPM. Yeah, so that's why they said that we shouldn't use that. This is the author's opinion. Okay. I think this is something good to know. And so the authors are proposing to model the counts directly without some normalization to make the data Gaussian-like. Probably you will argue that for this component, Gaussian-like is okay, but how do you deal with the zeros, right? Okay, now I want to use this quote from this paper. It provides another way to look at PCA, which is based on the Gaussian distribution assumption. So if you're interested in learning more about this, there is something you can search for. It's called probabilistic PCA. Probabilistic PCA just offers a model perspective using a probabilistic model to explain PCA. And then it has its own PC algorithm from that modeling approach. So this explanation is assuming Gaussian distribution. Although the way we explain PCA using the eigen decomposition of X transpose X is just purely linear algebra. We didn't make any probabilistic assumption over there, but this is the probability assumption approach. So PCA minimizes the mean square error between the data and the low rank representation, okay? By low rank, it means if we have K PCs, then the low rank is K, or embedding. Embedding is actually the PC score. Remember, we have every cell with a score in every PC. That's what we call the cell's PC embedding. Let Yij be the raw count and Zij be the normalized and transformed version of Yij. So this is like what Seurat uses, such as centered and scale log CPM, right? It's just like now Seurat changes to log CP10K, but otherwise it's the same thing, Z-score. The PC objective function is this. It tries to find the U vector and J vector. So Ui and Vj are both L dimensional. So L here is the number of PC. Previously, I said this, I called this K, but here the author used L. And Ui is what? Ui is the PC scores of cell I. Vj is gene J's loadings in the LPCs. So this is like a loading vector for gene J. This is the PC score vector for cell I. And this is something transpose. The prime here means transpose. So then you have an L dimensional vector transpose times an L dimensional vector. Linear algebra will give you a scalar. And the scalar is what PCA's reconstruction is. So you can reconstruct your original Zij. So Zij is cell I's original score in gene J. And we're reconstructing it using this inner product of these two L dimensional vectors. So this is another way to understand PCA. It's exactly equivalent to what we have before. It's just another matrix form to represent the same problem. So previously, we just write the matrix form, remember? So we can decompose the matrix, say Z, right, into a PC score matrix times a loading matrix. That's what we had before. But now we're writing it out element-wise. Okay, Ui are called factors, the scores we're talking, and controls the complexity of the model. Minimizing the MSE is equivalent to minimizing the Euclidean distance metric between the embedding and the data. So they call this embedding, they call this data. It's also equivalent to maximizing the likelihood of a Gaussian model. This is the new perspective we didn't cover because we talked about two things: maximizing the variance, minimizing the reconstruction error, and this is still minimizing the reconstruction error. But we can interpret it as the Gaussian model. So Zij follows a Gaussian distribution with this reconstruction part as its mean with some variance, sigma squared. So basically the equivalence here is that if you assume this Gaussian model and you want to do the maximum likelihood estimation for Ui and Vj, it turns out to be minimizing this MSE, mean square error. So they are equivalent, right? This is like the minimizing loss function approach. This is maximizing likelihood estimation approach. Yeah, and this equivalence is actually widespread in many machine learning algorithms nowadays. There is the NLE approach, and there is an equivalently minimizing loss approach. Okay, so without going into the derivation for this, because we can easily derive why maximizing the likelihood based on Gaussian is equivalent to this by writing out the likelihood. So for this, I also want to know how many of you have ever done maximum likelihood estimation for yourself. Okay, maybe only half of the class. I don't know if you actually took this 100B class, but I indeed strongly suggest to the bioinformatics program that for students who haven't had a background for this level mathematical statistics, introduction to mathematical statistics, I strongly recommend 100B. I think that's something essential. It's more important than 100A because 100A is just probability, but 100B is with the first class, offered in my department, statistics department at UCLA, to undergraduate students that really does mathematical statistics. So by mathematical statistics, in the process, you need to do some derivation, not just programming. Because for some other classes, maybe you can rely on R package and run it. But for this class, if you really want to understand many things, like the equivalence between this and this, you need some derivation. Okay, so this is the normal Gaussian model, so you can interpret PCA. Then I want to further say, how do we connect PCA for linear model, for Gaussian model, to the generalized linear model, to the GLM PCA for generalized linear model? So that's why I want to spend some time on the linear model versus generalized linear model. Maybe we cannot finish today, but we'll continue next time. So in the linear model, this is the simple linear regression. I think many of you should have seen, right? The response yi is assumed to be an intercept plus a slope times one predictor plus random noise. And this is for one observation, the ith observation, and the predictor xi is the value of the predictor in the ith observation. Yi is the value of the response in the ith observation, and this is the error term. So the key assumptions in simple linear model is that you assume the error terms, the noise terms are all independent of each other, and they are all, we call this IID, which stands for independent and identically distributed, which is a very standard way to say they all follow the same distribution and they're all independent from this normal distribution with mean zero common variance. So this normal is called normality assumption, and the common variance in the linear regression framework is called homoscedasticity assumption. So they have the same variance. Okay, this is the simple linear model. Now, if we want to make it vector form. So once you go beyond simple linear regression, matrix will make things much simpler. So now we stack the n response into a y vector, n-dimensional. We stack the n values of the predictor along with the column ones for intercept into a matrix. And this is called design matrix. And we have the two parameters, intercept and slope, stacked in the vector. And we have the error terms and error terms stacked in the vector. So using those notations, we can write a matrix form as y equals x times beta plus epsilon. So here the beta is two-dimensional, x is n by 2. So you can just simply do the dimensionality matching. You will see they're all n by 1 in each term. And in particular, for this design matrix, from now on, we will denote the ith row as xi transpose, which is two-dimensional here because it includes one term for the intercept, one for the predictor value. Okay, this is for simple linear regression. Now, multiple regression. When you have more than one features or more than one predictors. So in linear model, we often say predictors, but they are just the features, the same thing. So now we can have the design matrix as n by p. So the first column is still 1. And the other columns are other p-1 columns correspond to the p-1 features. And we still denote each row as xi transpose. So that's what we have. So here's the key thing. So we can interpret this linear model in a two-layer structure. For the systematic structure, it's about the mean of yi. So no randomness, just how the mean of yi can be explained by my p-1 predictors or features. So this is the linear part, the mean part. And typically, we give a notation for the mean of yi as mu I, but this is not the most important thing. You just remember this is the mean expectation of yi as xi transpose beta. So we assume that expectation of yi is just due to my predictors. And it's the predictors linear combination that explains the mean. Random structure is about the observed yi, how it is related to the mean of yi. So for linear model, we are essentially assuming that the yi is a normal distribution, mean is the systematic structure, xi transpose beta, and the variance is sigma squared. Actually, this is a typo, actually. This one should have this gone because this was a previous lecture notes without very careful proofreading. So that's why we have this problem. Yeah, but just let me see whether I can move it back. No, okay, yeah. So this in should be gone. So this should be gone, just sigma squared. It should be a scalar. But this one is right. This one is correct because in is just the n-dimensional identity matrix. So when we have this epsilon jointly, then it should be an n-dimensional normal distribution. While here it's just one-dimensional, so it should just be sigma squared. Yeah. All right, and this is a linear model. And the reason I want to introduce this is that now we will move on to generalized linear model. So then I think this can help you better understand PCA. Probably I will just go this slide and one more slide, then we'll be done. So for generalized linear model, it was motivated by the fact that your yi's, the responses are no longer continuous or real value. Real value means that it can be positive, negative with many digits, right? That's what our yi is because from a Gaussian normal distribution, the values of yi should be real value. So what if your yi is like binary? This can happen in many cases. For example, if you have the voting data, each voter would only vote yes or no. So that's binary. And if you want to predict this or explain the data using the voters' predictors like age, education, salary to explain their voting pattern, then this becomes a regression problem, but you don't have the continuous yi. Then what do? Right? So again, this two-layer explanation will make it feasible. So first of all, it makes sense to assume each yi just to follow a Bernoulli distribution because Bernoulli is the distribution that gave us binaries, right? 0 and 1 has a probability pi i. Pi I is basically the chance of getting 1 instead of 0. 1 minus pi I is the chance of getting 0. So from the property of Bernoulli, we know the expectation of yi is just pi I. Variance of yi is just pi I times 1 pi. This is just a property of Bernoulli. Then the question is, how do we link this expectation of yi to predictors? That's our question. Previously, in the linear model, we linked the E of yi to predictors using the linear function, xi transpose beta. But now, the EYI, expectation of yi is pi I, whose range is between 0 and 1. Then assuming this 0 to 1 value to be a linear function of predictors may not be possible. The reason is, if you think about this application, age, education, salary, if you want to encode each one as a value, like for age, it's a numerical value itself, right? And salary, it has numerical value itself. But if you combine them in a linear way, it's very likely the combination is not in the range of 0 to 1. Then how can you link that to pi I? The pi I is here, the mean. So that's where generalized linear model help us. So in particular, this is what we have. Maybe we'll continue, go back to here next time, but I'll just show you the end result. The end result is that you will do a logic transformation of pi y. logic function will change pi I from 0 to 1 to negative infinity to positive infinity. And then you assume logic pi I to be a linear function of your predictors. So this is what the logic function looks like. So when the pi I is 0.5, logic pi is 0. When the pi is going down to 0, logic pi goes down to negative infinity. When the pi I goes to 1, logic pi goes up to positive infinity. So it's a monotone transformation, but in the x range is 0 to 1, y range is negative infinity to positive infinity. So you do this transformation, and you are trying to assume the transform pi I, logic pi I is a linear function. Yeah, so this is the example for logistic regression. We'll come back to give you more detail. And for Poisson regression, we have another distribution for the random structure and the systematic structure. So because of this, we could generalize PCA to other distributions. So the idea is similar. So here PCA for normal, then we have something else for non-normal. And that's the idea of generalized PCA. Yeah. So I think the author just tried to remove the step for normalizing the data and directly work with data. Okay. All right, so that's all for today, and then I'll see you on Monday.